Hello everyone, welcome back to AS Biology with Dr. Demi. I am Dr. Demi, and in today's video I will be continuing with chapter 7 of the AS Biology syllabus. So if you haven't watched the first part of this chapter, which is chapter 7.1, I suggest that you do so because in that chapter, or in that video rather, I discuss in detail how water is transported through the xylem. In this video, I will be discussing how water is transported through the phloem and basically what is transported in the phloem altogether. So please pay attention and make sure you catch up on the previous video if you haven't watched that. So when we speak about translocation, or rather transport in the phloem, the word translocation is often used. And translocation is simply the movement of substances from one place to another, but it is used to refer to the movement of soluble organic substances. So things like sugars that are made from photosynthesis in the plant are translocated. So they're not really spoken of in terms of like just transportation, but I often tell students that another word for translocation is simply transportation. These molecules, like your sucrose that is made from photosynthesis, is called an assimilate. So you will see when you study this chapter that you will come across the word assimilates quite a lot. Assimilates will be transported through what we call the sieve elements, and sieve elements are found in the phloem tissue as well as other types of cells. So if you look here at this image, I'm just going to use my pen over here so that you're able to see. So this is the xylem, which is the one we discussed earlier in the previous chapter, and we said that the xylem is simply a collection of hollow tubes through which water is able to be transported. On the other hand, we have the phloem. The phloem is not a hollow tube. As a matter of fact, at the end of each phloem section, we have these little plates here that are called the sieve plates. And what these sieve plates do is they simply regulate the chemicals or the ions and minerals that move across. And we also said with the xylem that water moves upwards from the roots of the xylem to the other parts, whereas in the phloem the sugars or the sucrose would move from the leaves to the other parts. We call the part where the sucrose moves from, which is where the sucrose is made, we call that part the source, and we call the part where the sucrose will be stored the sink. So if you were to think about it in terms of a potato plant, like a sweet potato plant, the leaves of the plant would be the source because that is where photosynthesis happens and where sucrose is made, and then the roots where the sucrose is stored, which is what we eat, would be called the sink. So always remember the difference between source and sink. Something else to know about phloem tissue is that they have these cells called companion cells, and you will see why these become important as we start to discuss translocation in the phloem. The companion cells are alive, they are metabolically active cells, and they contribute to the uploading or the loading rather of sucrose within the cell. So, phloem is the main vessel, and it is usually used to translocate mineral ions as well as to translocate sucrose. Phloem cells are also called sieve tubes, and these tubes again consist of living cells. Remember that when we discussed xylem, we said that the xylem is made up of dead cells. It is not metabolically active, and water is simply transported through the xylem through the forces of cohesion and adhesion. In this case, it is a different story. The sieve tubes, which are the phloem tubes in this case, are made up of many sieve elements that are joined from end to end. So remember when we discussed the xylem, we had said that the xylem is a hollow tube made from hollow cells, or hollow pipes rather, that are just joined from end to end. It is the same with the phloem, except the pipes are not hollow. You also need to know that the sieve elements, again, which make up the sieve tubes, have cellular cell walls, they have cell surface membranes, they have cytoplasm that contain endoplasmic reticulum, and they have mitochondria. So that already tells you again that these cells are metabolically active and so are not dead. Each sieve element, when you look at it, the sieve elements, by the way, again, are speaking about the components that make up the phloem. So we can call this section over here, if you look at this image, this section here can be a sieve element, that's another sieve element. Altogether, they would make up the phloem, and each one has at least one companion cell that is attached to it. And what happens is that the plasmodist matter would allow for exchange of material between the sieve elements and the companion cells. It is very important for you as a biology student to be able to label these diagrams, because I have seen exam questions whereby you are asked to label them. So this here, we call it a sieve plate. As you can see, it has holes in it, and so it serves as a sieve, just like the name suggests. So you should be able to know the sieve plate, you should be able to know the sieve tube element. So just one section of this is a sieve tube element, and you should be able to know the companion cell, which is the one attached to the sieve tube element. So when we speak of translocation, the big question is, how does translocation happen? How do molecules of sucrose move from one part of the plant to the other? Now remember when we were discussing movement in the xylem and we spoke about water movement, we said that the movement of water is dependent on the difference in water potential, which means that at the leaves where water is being lost, the water potential is usually lower than at the roots where water is being absorbed, and as a result of that, the water continues to move from the roots towards the leaves. And that is a form of passive transport because it is simply osmosis. And if you recall chapter 4, osmosis is a kind of passive transport. In phloem, it is a different story. Mass flow in the phloem, which is the flow of water and all the solutes in it as one body of fluid, involves active transport. And that is why when I was describing the phloem on the previous slide, I said that the companion cells of the phloem have endoplasmic reticulum, they also have mitochondria. So that is an indication that there is some kind of energy that is being used in the transportation of the sucrose in the phloem. Now, when we speak about the different parts of the plant, I also mentioned the source and the sink. So in the source is where you take the sucrose and load it into the phloem, while the sink is where you take the sucrose out of the phloem and store it. So always think of the source as the leaves, and you think of the sink as places like the stems and the roots. What happens then is that when sucrose is loaded into the phloem, and this is something to bear in mind, what do you think would happen to the water potential in the phloem? Obviously, this decreases the water potential because now you have put in a lot of sucrose into the phloem, or what we often call the sieve elements. And as a result of this, water will then flow into the phloem through osmosis. Remember again, when we were discussing xylem and the movement of water, we mentioned what we call the pits. And we said the pits allow movement of water out of the xylem tubes into neighboring xylem tubes. They also allow movement of water from the xylem tubes into surrounding cells that might need water. So in this case, when we have the loading of phloem into the loading of sucrose rather into the phloem, what happens then is that water would move into the phloem in order to ensure a mass flow through the sieve tubes. So this means that water also aids in the transportation of the phloem sap. Now you need to know that the phloem sap can flow up or down, it doesn't matter, unlike the xylem which only flows in one direction, upwards. But it is important to know that with the phloem, it only flows in one direction in any particular sieve tube. Now that's not very important for your exams. I just thought to put that in there in case you were interested. So, how is sucrose loaded? This is the big question. Now, we all know that in the leaves we have photosynthesis that happens, which means that the leaves of every plant, whenever there is sunlight, will open up, they'll take up CO2 and then undergo photosynthesis. Just by the way, photosynthesis is an A-level subject, A-level topic rather, so you would have to wait until your A-levels to fully understand that. But the end end product of photosynthesis is usually the sugars, and those sugars are stored in the form of sucrose. Now, sucrose needs to be transported from the leaves to other parts of the plant where it can be stored so that the leaves can be able to make more sucrose. What then happens in this case is that the sucrose is loaded into a companion cell. So I have done this outline over here for you. I'm just going to draw a red line here so that you see this is the outline that just tells you all that you need to know about sucrose loading. So sucrose is loaded into a companion cell. Remember that the companion cell is right next to the sieve element which makes up the phloem. Sometimes it can be loaded directly into a sieve element by active transport, but usually it is loaded through a companion cell. The reason why it is loaded by active transport, just by the way, is that the concentration of phloem, of sucrose rather, inside the phloem is always going to be higher than the concentration in the leaves because the phloem is constantly transporting sucrose. And so in order to get the sucrose out of the leaves, you need to use ATP. And so what happens here is that in terms of the companion cells, what will happen is that the cell membrane over here, you can look at this image over here. We call this the inside of the cell. So I'm just going to write inside. I don't do very well with writing with a mouse, but that's the inside, and over here is outside. Okay, so in order to transport sucrose into the phloem cell, what will happen is that there will be proton pumps on the surface of the cell membrane that will pump out protons into the cell wall, or the space between the cell wall and the cell membrane. That then causes the outside to become more positively charged than the inside. But obviously something that we know is that the body always tries to stay in a state of balance, and that applies to plants as well. So the proton ions that have been pumped out will have to come back in. Now, proton ions are unable to simply diffuse through the cell membrane. That doesn't happen. They have to come in through a transport protein. This transport protein in particular is called a co-transporter because it carries the hydrogen ions that were pumped out as well as sucrose, which means that for this transport protein to function fully, it will take a hydrogen ion and bind to it, but it also requires a sucrose molecule to also come through. And when those two come through, then the protein is able to open up and it brings in the hydrogen ions as well as the sucrose ions, and this then enables the sucrose to move from the companion cell into the sieve tube. So this is how sucrose is loaded through the companion cell. It's very easy for you to remember it if you're going to write this in your exams, to simply just follow the outline from here to there to say that the sucrose is loaded into a companion cell by active transport. Hydrogen ions are pumped out of the companion cell through its cell membrane to the space between the cell membrane and the cell wall. Those hydrogen ions will increase in concentration on the outside, and to create balance they would have to be brought back in. And so what happens is they come in through a co-transport protein which brings in hydrogen ions and sucrose molecules. When the sucrose molecules and hydrogen ions come into the companion cell, they can then move, the sucrose can then move into the phloem from there. The differences between the sieve tubes and the xylem vessels are questions. This makes a question that I totally enjoy seeing in the exam. Something you have to bear in mind: the sieve tubes are also called the phloem cells. Please don't forget that this is also phloem. It's just very common for it to be referred to as sieve tubes. So the phloem cells are made up of living cells. They don't have any lignified cell walls, and the end walls form sieve plates, which means that at the end of each phloem cell, you can see over there, let's assume this is the end, you will see that there are tiny holes here, and then that leads to the next one. This is a sieve plate, and the sieve plates simply allow the phloem to seal itself, so if it's damaged, so it simply repairs itself. The xylem vessels, on the other hand, are made up of dead cells rather. They have lignified cell walls that strengthen them. They don't have any end walls, and they don't have any sieve plates. So I have deliberately put these here side by side so that it is easy for you to compare. And why is it important that these differences should be known? If the phloem cells were dead, then there would be no way to actively transport them. That means then in that case that phloem would simply just leak out and it would be wasted. That means sucrose rather would leak out and it would be wasted. The xylem cells are empty, so they allow water to flow without any obstruction, which means that when water flows through the xylem, it doesn't have to deal with any metabolic processes or be interrupted by any cell membranes in its movement. The lignified cell walls of the xylem vessels, like I said earlier, help to support the plant. So when you look at a tree, trees are very sturdy because the xylem vessels in them also add to that strengthening. The sieve plates in the phloem tubes help to maintain pressure in the tubes, because when the sieve plates are there, then you're able to keep a certain section pressurized in order to facilitate transportation. And also sieve plates allow the phloem cells to quickly transport sap, quickly clot sap rather, so that there is no loss of sucrose, and also to reduce the vulnerability to diseases. So yeah, that's it on transport in plants. This is actually all that you need to know. What I have prepared in addition to this is a video about drawings. I'm just going to show you what the drawings should look like when you do your microscopy practical, so that you're able to just follow that. But other than that, this is the end of transport in plants. If you feel like you still don't understand anything, or there are things you need clarification on, please post a comment, and I will try to get back to you as soon as possible. Thank you so much for watching. Until the next video, have a good time.